Okay, hello, everyone. All right, a quick announcement before we get started. I got a couple questions about an old renal anatomy link that was showing up for you guys. That was from last year, which is why you didn't have a copy of that worksheet. But I looked back at it, and I think it would be good review for your exam. So it correlates to some of that stuff in the video lecture that you should have viewed already. So I'm putting that up as a 5-point extra credit assignment for you guys. So go ahead and finish that by Monday. You can find it linked here under Unit 3 or also under the assignments tab. Okay. So that's just making sure that you know some of these anatomical structures of the kidney. All right. That said, we are going to dive back in to our discussion of basically water reabsorption and how we control it. Okay, so thus far we've talked about the fact that we have a lot of water reabsorption. Let's trace through all our water actually. So we have water coming from your plasma being filtered out from the glomerulus, from that capillary bed. It's forming most of our tubular fluid when we start out. Lots and lots of it, dependent on the glomerular filtration rate. But most of that fluid that we're filtering is water. As we go through the proximal tubule, that first part of the nephron, we have lots of water reabsorption happening because otherwise we would be losing way too much fluid out of our blood. So we have water being transported from that tubular fluid back into the blood, specifically into our peritubular capillaries. And we traced through how that's following our sodium reabsorption in the proximal tubule. Okay. So we're moving along. We next go down into the loop of Henle, which is what we talked about for most of last time. So our proximal tubule was up in the cortex of the kidney. We had an osmolarity of 300 in the cortex. We had an osmolarity of 300 milliosmoles in our tubular fluid in the cortex. Then we start to dive down into the medulla of the kidney. So that's our renal pyramids. We start to have essentially a greater pull on water due to high solute concentration in that tissue. That high solute concentration is coming from active transport of our solutes, our ions, from the ascending limb of the loop of Henle. But if we're following the fluid, right, that fluid came from the glomerulus, filtered, plenty of it was reabsorbed in the proximal tubule. Now we're diving down into the descending limb of the loop of Henle. That descending limb is permeable to water. It's moving through an area where we have really high osmolarity, but it's starting out with its own osmolarity of just 300 milliosmoles. So we have water movement towards that high osmolarity of the medulla. Okay, so we start to concentrate the filtrate, starting to concentrate it into urine as we go down to that descending limb. Okay. And then we go back through the ascending limb. So we created a highly osmolar fluid as we got to the deepest parts of the medulla, but we've removed water. Now, as we go up through that ascending limb, no longer permeable to water, but now instead we have active transport of solutes. So we're taking those salts, essentially, actively pumping them using energy into the medulla, keeping that medulla nice and salty, and lowering our osmolarity of the fluid back down. By the time we exit that loop of Henle, exit that ascending limb, we're down to an osmolarity of 100 milliosmoles. That's our distal tubule that we're in now as we exit the loop of Henle. So it has a osmolarity of 100 milliosmoles. It's in the cortex now, which still has that baseline value of 300 milliosmoles. So there's a little bit of a pull on that water in the fluid traveling through the distal tubule. Okay, so if we have the correct aquaporins, we would have some water movement across that cell layer towards the cortex. Okay, and then we go down into the collecting duct. We haven't had any water movement. We're still at 100 milliosmoles in our fluid. We're diving down through the medulla. It has the same values as the medulla had when we were going through the loop of Henle. So it's getting more and more osmolar, creating a stronger and stronger pull on water as we go down. If we don't have the aquaporins that we need, that collecting duct isn't permeable to water, so nothing happens. But if we do have aquaporin 2 and aquaporin 3 present, that cell layer becomes permeable to water, which means that we start to pull water out of that fluid, put it back into our blood, reabsorb it. Basically, that's our word, right? Reabsorbing that water, concentrating the urine back down, okay? Creating highly concentrated urine and maintaining blood volume through that process. So that's a whole big picture of what we went through on whatever day that was, Monday? Are we only halfway through the week? Okay. So that's basically what I want you to know of everything we did on Monday, right? I talked for an hour, but that's what we were trying to understand. Okay. So when we left off, we were thinking about that last piece, that distal tubule and collecting duct reabsorption. Okay. So we had our cells, right? And we were looking at whether or not they're permeable to water. So we have our basolateral membrane. We have our apical membrane. Apical membrane is facing the filtrate, so facing what's eventually going to become your urine. Basolateral membrane faces your blood. We have our 2 aquaporins potentially. So aquaporin 3, always present here on the basolateral membrane. Aquaporin 3. And we had our aquaporin 2, which may or may not be present on our apical membrane. So that's what we're varying to determine whether we can reabsorb this water. Okay. So we consider this regulated reabsorption because this whole process is going to be controlled by our hormone ADH, antidiuretic hormone. Okay. So antidiuretic hormone is what's controlling whether or not we have this aquaporin 2. Okay. So in the presence of ADH, okay, we are going to insert this aquaporin 2 on the apical membrane, allowing us to reabsorb more water. Okay. And so to remember that, right, the way I like to do it, right, I like to break apart that word. I'll write it out. Okay. Okay. So it's a hormone, which tells us it's about cell signaling, part of our endocrine system, going through our bloodstream, something far away. Okay. And about its function, we can look at this antidiuretic part. So this is going to depend on whether you've thought about diuretics before in your life or not. Okay. But if you eat or drink something that's a diuretic, basically that means it makes you pee a bunch. Okay. So diuretics would create high volume urine. And the way we can have high volume urine is it would have a lot of volume, a lot of water in it, right? So it would be dilute, it would be high volume. That's what a diuretic is. So if you have a medicine that you take that's a diuretic, that's what's going on. So antidiuretic hormone fights against that. Okay. So the diuretic is making you pee a lot, high volume. You want to fight against that antidiuretic. You want to have less volume in the urine. The way you would control that, right, is going to be through how much reabsorption happens. So reabsorption okay. Reabsorption fights against that excretion of lots of water, essentially. So that's how I kind of work through remembering which direction ADH is working on. Okay. So you might also think about that equation we had for relating excretion to reabsorption and secretion as well. So you could kind of map out why reabsorption creates less excretion that way as well. Okay. So ADH is controlling whether we have this aquaporin 2 on our apical membrane. Okay. So this is what creates a range of osmolarity in the urine, because they're controlling how much water gets reabsorbed. If you reabsorb more water, essentially you're concentrating solutes in the urine. So the more water you reabsorb, the lower the volume of the urine is going to be, and the more highly osmolar it will be. Right. So osmolarity isn't a direct measure of solute concentration, but if we want to like wave our hands and think of it that way, it's ballpark right? So the higher the number, the more concentrated solutes you have in there, okay? All right, so we're going to walk through this process, but that's our big picture view. We're going to go piece by piece. Okay. So we're going to start out, here we have our nice little cell, and specifically what we're looking at is something called a principal cell. So our principal cells are lining parts of the late distal tubule and the collecting duct. And this is where we can have this regulated reabsorption happening. Okay. So in order to have more reabsorption, so to get more water back out of your urine and back into your blood, okay, you need ADH floating around in your plasma. So the ADH is going to be coming from somewhere else. Right, this is a hormone. Specifically, it's going to be coming from your posterior pituitary. So we'll get there in a bit. Okay, it's going into your bloodstream. Okay, so think back to our endocrine system, and it's going to seep out of our bloodstream and find itself some receptors on our principal cells. Okay. So here we see that ADH molecule coming in and binding to our ADH receptor on our basolateral membrane of this principal cell. Okay, so this is how we receive that signal. So how we know that we have ADH present, so we know we're going to change how much water we're reabsorbing. So the ADH receptor happens to be a G protein coupled receptor. So we can see that G protein next to it. So that was one of our types of receptors. So what happens next is that G protein dissociates. Part of it scoots along the membrane and binds into another enzyme. Okay. And this enzyme then triggers the creation of one of our secondary signaling molecules, cyclic AMP. Okay. So next we're making cyclic AMP, which controls a lot of processes in our cells, right? We've seen that molecule before, like when we were looking at our different types of adrenergic receptors, okay? So this is just a common secondary messenger system molecule. All right, so when we make that cyclic AMP, what it does next is it helps us create a molecule called protein kinase A. We don't need to focus on that part, but there's a whole signaling pathway inside, essentially. Ultimately, what we're doing is we're taking little vesicles of aquaporin 2. So those are this light blue molecule here is our aquaporin 2. You can see it labeled here. And in response to the ADH, ultimately what we're doing is we're inserting those into the membrane. So this vesicle would touch the membrane, burst open, and then suddenly those additional aquaporin 2s have merged into our membrane. So we can see here that we have lots of aquaporin 2 molecules where this vesicle attached to it. Okay. The function of that aquaporin 2 molecule, or this aquaporin 2 is specifically a type of protein, right, is to allow water into the cell. Okay. So we're reabsorbing water here, right? So we're reabsorbing, so we're pulling it out of the filtrate, concentrating down that filtrate into urine. Okay. Goes through aquaporin 2 first, and then it goes through aquaporin 3 second. Aquaporin 3 is always present on the basolateral membrane. We don't need to worry about whether that's there or not. And then that can passively cross into our plasma, giving us nice high volume plasma, keeping that water, keeping us hydrated, flowing through our body. questions about this process here? From this diagram, the stuff you want to be focusing on, right, is knowing that this is happening in the presence of ADH. You want to know that we're triggering this insertion of the aquaporin 2 on this apical membrane. And you want to know that this is allowing us to transport water all the way through back to the plasma. This is reabsorption. And then you would also want to be able to think about what's this doing to the amount and concentration of your urine when you're reabsorbing versus not reabsorbing. Okay. But this is the whole mechanism of action painted out for us. Okay. So we do also regulate whether we are secreting this hormone. Right? So our antidiuretic hormone, we only want to be reabsorbing water sometimes, right? We want to reabsorb different amounts depending on our situation. Right, so it's good to stay hydrated. It's good to keep water in your blood, in your plasma, but too much is also a problem, right? We've thought, as we think about filtration pressures, how that hydrostatic pressure is the pressure of a fluid on the walls of its container, right? So if we have really high volume plasma because it has a really high amount of water in it, right? This would create stress on the walls of a blood vessel, essentially, and change those filtration pressures, which is not always good, right? We want to do homeostasis. We want to do hemostasis, which is just our version for like maintaining blood, right? So we want to keep this environment consistent. We don't want to have huge variation in concentrations of things. We don't want to have huge variation in volumes of things. And I guess by things here, I really mean our plasma, right? We know that lots of systems in our body are dependent on the concentrations across our membranes, right? If you think about our neurons, right? That was really driven by knowing what direction the sodium is going to flow, knowing what direction the potassium is going to flow. If we changed the concentration of the surrounding environment, that might change, right? In which case we would affect how your nerves function. So that wouldn't be too good for us, okay? So we want to maintain a consistent environment. Okay. So we have triggers for the secretion of ADH. So ADH is coming from our posterior pituitary. Okay. So we remember maybe way back when we had our pituitary kind of hanging down from our brain, sitting in that sella turcica. We had an anterior and a posterior part. We talked a lot about the anterior pituitary hormones. We had tons and tons of them. Now we are finally talking about a posterior pituitary hormone. So a really important posterior pituitary hormone is ADH. Okay. And those hormones in the posterior pituitary were functioning in a different way. We had a whole portal system going on in the anterior pituitary, but in the posterior pituitary, we just have cells in our hypothalamus that extend down into our posterior pituitary, and they release our hormones, in this case, ADH. Okay. So that's where the ADH is coming from. So that's then going to get into your blood system. It's going to travel all around. It's going to travel to your kidney, which is where we are getting the signal. Okay. Now, in order to determine whether we want to release it or not, okay, we do have an organ essentially that senses osmolarity. So osmoreceptors and something called the organum vasculosum of the laminae terminalis, or OVLT, which always looks like Ovaltine to me, senses osmolarity. So what it's doing essentially is it's sensing, right, like how salty your blood is, right? It's sensing the concentration of solutes, essentially. And since we want to maintain that concentration of solutes in your blood plasma at a certain level, right? Those osmoreceptors, by sensing that, they're going to know, do you need more water to water down these solutes that we have? Or are you good? Can you keep just excreting your water, right? So those osmoreceptors are sensing how much we want to reabsorb additional fluid, okay? So this region where we have these osmoreceptors isn't surrounded by the blood-brain barrier, which is part of why we can sense this. Okay. So that's one way that we're sensing whether or not we want to reabsorb water. If we want to reabsorb water, we need ADH. Okay. So if those osmoreceptors are sensing high osmolarity, they're going to want to water that down, which means we're going to need to reabsorb water, which means we need to release ADH. Okay. So the other thing that's going to tell us maybe we need to release some ADH is our baroreceptors. Okay. So the baroreceptors we talked about when we were talking about maintaining blood pressure, right? So these are sensing blood volume, blood pressure, right? We had some in our arteries, we had some in our veins. Okay? So when we sense changes in blood pressure, specifically if that baroreceptor activity is going down, or sensing a fall in blood pressure, okay, then we're going to want to reabsorb more water to get that blood pressure back up. Because if we have more volume in our cardiovascular system, that's a higher pressure, right? Just like if you have a water balloon, more water in it, higher pressure, or like a hose, more volume going through, higher pressure. So those are like our two signals here. So osmolarity and blood volume / pressure. Okay. Both of those can trigger the secretion of ADH because both of those are problems we could fix by absorbing more water. So we have some little diagrams of basically what I just said to you out loud. Okay. So this first one is showing us what happens if we have an increase in the osmolarity of the extracellular fluid. Okay. So we're saying the extracellular fluid near these receptors is getting pretty salty. We're sensing like highly concentrated plasma, which has then created highly concentrated extracellular fluid. Okay, so our osmoreceptors, okay, are going to detect and respond. They're in the hypothalamus, so that part of the brain. The hypothalamus contains neurons. Their activity is increasing. The effect of those neurons is to release more ADH. Okay. When we release that additional ADH, circulating around your bloodstream until it gets to your kidney. When it gets to your kidney, it says, okay, let's insert more aquaporin 2 into that apical membrane so we can reabsorb more water. Okay, so we reabsorb more water at the kidney, which means we excrete less water because these are in opposition to each other, right? If you're reabsorbing water, you're not getting rid of water. And vice versa, if you're not reabsorbing water, it's got to go somewhere, you'd excrete it. Right? But we're reabsorbing more water because we have ADH. So we decrease how much water we're excreting, so how much water you're peeing out, which means you're conserving how much water is staying in your body and therefore staying in your plasma, hopefully watering down that osmolarity in the extracellular fluid. So this is a negative feedback loop. So then after this happens, we could reverse all these arrow directions, right? So that would get us back to like our steady state. Okay. So the other piece that's happening is a reaction to blood volume, essentially. Right? So this is how we can long-term help regulate blood pressure by regulating how much physical blood you have. Okay. So in this case, we would sense a decrease in blood volume in the veins, which are like volume holders for blood. And we would also be sensing a decrease in mean arterial pressure at the same time, right? If you have not very much blood, you're going to have not very much pressure. Okay. So both of those things go together. So our baroreceptors are pressure sensors, essentially. They detect and respond, and then they go tell the hypothalamus. So the hypothalamus says, okay, we don't have enough blood, our pressure's dropping. Let's keep as much water in our blood as we can. So this part is the same. So we're going to increase the activity of those cells in the hypothalamus, which increases how much ADH they secrete, which tells the kidneys, okay, make that aquaporin 2 so we can reabsorb more water. We're not getting rid of our water anymore, not excreting it. And we're keeping as much water in the blood as we can in a negative feedback loop until we're back to a good amount of blood pressure, a good amount of blood volume. Okay. So those are the two pieces that are going to influence how much water we reabsorb, mediated, so affected by this antidiuretic hormone in the middle. That's our signal. So another way that water excretion does get kind of regulated, if we're thinking big picture, is by changes in the glomerular filtration rate. Okay, so usually we have auto-regulation of that glomerular filtration rate. Okay, so usually we're in this plateau where we have a glomerular filtration rate of about 125 milliliters per minute. And we really like to stay in that range regardless of our mean arterial pressure. But if we have too low blood pressure or too high blood pressure, we're going to overcome those mechanisms for regulation. So we had our myogenic regulation, essentially the smooth muscle, right, of our arterioles being able to clamp down, change resistance, change blood flow to the kidney, therefore help us change our glomerular filtration rate. So that changes our hydrostatic pressure in the glomerular capillary. We had our tubuloglomerular feedback. So as our nephron loops back around, that fluid essentially in our late distal tubule is kind of touching the cells in our renal corpuscle, that glomerulus. So we can get a signal there as well to change glomerular filtration rates. We also had mesangial cells, which are kind of covering over the glomerular capillary, being able to change how permeable that membrane is by contracting. So those were how we were staying here in this central range. Okay, so we had a slide on that, basically, is what I'm telling you. Okay. But otherwise, if we're outside of this range, we are going to have some overall problems. Okay. So if we are below a mean arterial pressure of about 80, it's real low. Okay. So overall here, this means we're not going to have enough pressure into the glomerular capillary, right? So we're essentially not filtering enough water because that hydrostatic pressure of the glomerular capillary, so PGC, was our main force for glomerular filtration, right? That was the biggest one for creating filtration, creating our glomerular filtration pressure, therefore creating our glomerular filtration rate. So if you don't have enough pressure in the capillary, okay, if you drop this number down too far, you don't have enough filtration, okay? So if you don't have enough filtration into the nephron in the first place, there's not really enough water left for you to excrete. Okay, so a really low glomerular filtration rate is a reaction in part to an extreme drop in arterial pressure because that arterial pressure affects the pressure of the glomerular capillary. So that's another reason you might see changes to the excretion of water from the kidney. Okay. On the opposite side, if we're like another color, we're up here, right? So if we have a really high mean arterial pressure, okay, this is forcing blood real hard through your cardiovascular system, right? High blood pressure. Okay. This means you're going to have a really high pressure in the capillary. So high pressure in the glomerular capillary, because the glomerular capillary just is part of your cardiovascular system. And this is too high for our regulatory mechanisms to kind of compensate. Okay. So in this case, we have a really high hydrostatic pressure in the glomerular capillary, which means we're going to have lots of water flowing out of that glomerular, those glomerular capillaries, right? Because essentially, right, like it's spraying out all these holes, right, real hard because we have high pressure. Okay, so we'd have an increase in the water filtering into the nephron. So if there's lots and lots of water moving really, really fast, you're going to end up peeing a lot. You're going to increase water excretion, just partially due to how fast that's moving, how much filtrate there is, okay? So this would only be occurring in pathological circumstances. It would be pathological to have a pressure that high, essentially. We should be able to regulate our mean arterial pressure better than this, and we should be able, therefore, to regulate the glomerular filtration rate. So these are like two additional reasons you might see changes to water excretion that aren't related to ADH. Right? That's how that kind of like links up. So these are just some questions that you can practice on later on. So we're going to move forward and talk about regulation of sodium and potassium now. And really, we're going to be regulating the two together for the most part. So we're going to focus a lot on sodium because we always like to focus a lot on sodium. But as we're regulating sodium, we're kind of doing potassium alongside, right? And this is always because many of these things, right, are driven by those sodium potassium pumps. Sodium and potassium go in opposite directions. So if you do something to the sodium, you're often affecting the potassium as well. All right, so we're going to be looking at two new hormones. I'm sorry that all our hormones in this unit start with A, but now we're going to be looking at aldosterone and A&P, atrial natriuretic peptide. Okay. These are going to be the two hormones that are affecting our plasma sodium levels. So ADH for water, aldosterone, and A&P for salts, essentially. Okay. All right. So we're going to start with thinking about the sodium. So first, some words, some vocabulary terms. So when we are talking about how much sodium you have in your blood, you have some specific vocabulary terms for this. So high plasma sodium, we call hypernatremia. So emia is our root for blood. We have this Na here for our sodium, right, which is Na plus. Okay. And hyper, if you're really hyper, you're really extra, means a lot. Okay, so what we're saying here is a lot of sodium in your blood. That's all that word means. Okay, so hypernatremia. So we can break down the opposite as well. So hypo means low. So low sodium blood would be hyponatremia. Okay, so these are just two terms we use to determine or like to describe whether someone has high plasma or low plasma sodium. So these are things you might measure in a blood test, basically. Okay. Now, how much sodium you have in your plasma is important because plasma is an easily sampled form of extracellular fluid. Extracellular because it's like outside all your blood cells, right? It's going to have concentrations similar to extracellular fluid like surrounding your tissue. Okay. So we can sense it easily by taking a blood draw, and those concentrations are really important because concentration gradients across membranes are what drives which directions active transport, which directions passive transport. Where are we going to have a force for movement? Where is our electrochemical driving force going, right? So if we change the concentration of solutes, and specifically of sodium in our extracellular fluid, suddenly all these physiological processes that we've been going through are going to get really shifted. They're going to get really wonky because we aren't going to have the same forces anymore. Okay, so sodium is critical for normal osmotic pressure. Okay, so it's critical for the normal force, that pull on blood. Okay, and it's also critical to functioning of excitable cells. So excitable cells are the ones that have action potential. So we're thinking nerves, neurons, your brain, and we're also thinking like heart cells, right? So sodium and solute concentrations are actually really important when we're thinking about heart problems as well, potentially. So we are going to overall want to be able to control how much sodium we're reabsorbing. We're going to want to toggle it. But sodium is one of the things that's getting freely filtered out of the glomerulus. right? So like everything that's in the plasma, except for our proteins, essentially, is just coming into the nephron from the glomerulus, including sodium. Okay. So we're having a lot of sodium come through. So similar to water, we're going to have reabsorption in the proximal tubules. We're going to have a lot of reabsorption in the proximal tubules. This is our our mass reabsorber. Okay. And then we're going to have more reabsorption in the distal tubules and collecting ducts, but there we're going to be regulating essentially how much sodium we're reabsorbing. Okay. There's no secretion of sodium. So secretion is the opposite direction from reabsorption. So secretion would be like if you're taking it out of the blood and putting it into our filtrate to become urine. That doesn't happen with sodium. Okay. So we only have to worry about this reabsorption direction. So our regulatory hormones here are going to be aldosterone and ANP. And we're going to focus more on the aldosterone. The ANP is like an extra. Okay. And we're going to be regulating it in those same cells where we're regulating our water reabsorption. So we're going to be targeting those principal cells in the distal tubules and the collecting ducts. Okay. Overall, we're going to need energy to do this process. So this is going to be active reabsorption. Okay. And like most things, it's going to be driven by the sodium-potassium pump. Really easy to remember here, hopefully, because we are literally talking about sodium, which is in the name of this pump. Okay. Okay. So this is just a quick reminder of what was happening in the proximal tubule. So we traced through this while we were thinking about why and how we're absorbing glucose. But if we're paying attention to sodium specifically, also clearly relevant. Okay. So we had our active transport of sodium and potassium by the sodium-potassium pump here at the basolateral membrane. So they're going opposite directions. We're creating a low sodium environment in this cell. This is a proximal tubule epithelial cell, so up at the front of our nephron. And so when I say this is low, I mean on either side, we have relatively higher sodium. So high sodium and relatively higher sodium here in our tubular fluid. Okay. This allows us to passively transport sodium into the cell, although we are usually using it to drive some secondary active transport. Right? This molecule X is probably glucose. Okay. Then we actively pump it out the basolateral membrane. And then because we have a high concentration here in the peritubular fluid, it can go into the plasma passively. All right. So that is our unregulated, essentially, sodium reabsorption in the proximal tube. All right, so no hormones involved here. Okay. Distal tubule is going to be different. Okay. First, we're just going to look at our transporters involved. So on the basolateral membrane, we're still going to have a sodium-potassium pump. Right, so we're still creating a low sodium environment here in the principal cell. We're still going to be doing just passive transport into our plasma on this side. But what's going on the apical membrane is a little different. So specifically, we're going to have channels for both potassium and sodium here on our apical membrane. Okay. So we have channels for just sodium to flow through. Changing whether we have these channels, like changing our aquaporin 2 presence, is going to change how much sodium we can reabsorb in our distal tubule cells. Okay. So these, we're going to be changing how much of them we have. Okay. All right. So overall, when we're thinking about aldosterone, just like our antidiuretic hormone, our ADH, increased water reabsorption, aldosterone is going to increase sodium reabsorption. Okay. So both of these are increasing our main thing that we're thinking about. Okay. Just now we're thinking about sodium. So instead of coming from the hypothalamus and the posterior pituitary, aldosterone is actually coming from your adrenal cortex. So remember our kidney, all right, here has a little hat, our adrenal gland. adrenal on top of the kidney renal. Okay, so steroid hormone up here, aldosterone, is just coming down and affecting the sodium reabsorption in your kidney pretty directly. It's affecting those principal cells that we were just looking at in the distal tubule and the collecting duct. What it's doing, mainly, the biggest effect is opening these sodium and potassium channels. It does also change how hard, how fast you're pumping sodium out of the cell and potassium into the cell. So what that can do is, instead of just having like low sodium inside the cell, we can make really low sodium by pumping out even more, meaning that we have a stronger force for sodium to come into the cell. If you have a stronger force and you have a lot of open channels, it can come in real quick. Yeah. So that's the overall effect of the aldosterone. So we can see this traced out the way we traced out the ADH. So if you have aldosterone in your blood, it's going to get into the cell. Aldosterone is a steroid hormone. So actually, it doesn't need a receptor on the plasma membrane. So aldosterone is able to diffuse across that membrane because of its physical and chemical properties. So think about how like cholesterol is inserted in the plasma membrane. Cholesterol was a steroid, essentially, the basis for forming steroids. So aldosterone can actually just cross through our plasma membrane. It's small enough and it doesn't have a charge, so it can go across. All right. So it does have receptors, but they're on the inside of the cell. Okay. It does these two things. So it's making us more of these channels, and it's keeping the sodium concentration inside the cell really low because it's speeding up how fast we pump that sodium out by changing the number of these channels, or sorry, changing the number of these pumps. Now, unfortunately, there's a big pathway for how we actually end up releasing aldosterone. So there are a number of different things that kind of get converted in order to release aldosterone from that adrenal gland. Okay, so this figure at the end shows us aldosterone in the blood coming from our adrenal cortex. Okay, but you can see that there's a lot going on first. Okay, so this is our renin angiotensin aldosterone system, or RAAS for short. Okay. So this RAAS system is really important to releasing aldosterone and therefore helping us maintain solute concentrations by regulating how much sodium we're reabsorbing. And I believe that this is affected by many disease processes. I've definitely seen some of the graduate students writing out in detail all these various steps. Okay, so we're gonna think about this RAAS system. All right, so this is going to start with the secretion of renin. Okay, well, at least from our perspective right here. So we're going to be secreting renin. So this is coming from the granular cells in the juxtaglomerular apparatus. Okay, so I was just mentioning part of our juxtaglomerular apparatus as we were thinking about or remembering the regulation of our glomerular filtration rate. So our glomerular filtration rate had to do with our macula densa cells. I'm just going to write, that was GFR. Okay. Our mesangial cells were helping us regulate GFR. Okay. So our macula densa cells were here sensing what's coming up through our nephron loop and going into the distal tubule. Our mesangial cells were controlling basically contraction kind of around the glomerulus. All right, now we're thinking about the granular cells. So the granular cells here are the ones that are secreting renin. So you can see these granular cells here tucked into this space between our end of the nephron loop and our afferent arterial. And we can see some on the other side of the afferent arterial as well. Okay. So what that renin is doing is that renin is going to be affecting a molecule that we already have in the blood that came from the liver. So probably all having flashbacks to our clotting pathways right now, or if you're not, you should, because we have something called angiotensinogen coming from the liver, swirling around in our bloodstream all the time, just like we had those inactive clotting factors coming from our liver, swimming around in your blood all the time. So angiotensinogen, whenever we see that inogen ending, that's kind of a signal to us that this is something that's not active yet, but needs to get converted. Okay. So renin converts this angiotensinogen to angiotensin 1, right? Then we have another conversion process. So there are two angiotensins, angiotensin 1 and angiotensin 2. So you go from 1 to 2, at least the numbers are in order here, unlike with our clotting factors. And the way we get from angiotensin 1 to angiotensin 2 is something called angiotensin converting enzyme. Okay, so converts angiotensin 1 to 2. So angiotensin converting enzyme, or ACE. Okay, so this is embedded in the walls of the capillary. Okay, so your angiotensinogen coming from your liver combines with your renin, makes angiotensin 1. Angiotensin 1 bumps into the wall of the capillary where we have this angiotensin converting enzyme, becomes angiotensin 2. And that is what signals to your adrenal gland to produce aldosterone. Okay. So big, long, well, I don't know, for physiology, maybe not actually that long, but we have this pathway of molecules leading to this creation of aldosterone, which then gets to your kidney, tells you to reabsorb your sodium. Okay, so this is just a big picture of it. Okay. And we have some pathways we'll pick up here next time. Okay. And basically, the same thing is happening to the potassium, but in the opposite direction.